Welcome, everyone, and thank you so much for joining us today. I'm Tiffany Chen, the Chief Medical Officer here at Techcyte, and I'll be your host today for the next half hour. Today's session is part of our ongoing webinar series, Pioneers in Digital Pathology. And our session today is entitled Deploying AI-ready IHC Biomarkers in Digital Pathology Workflows. So a bit of housekeeping before we start. Feel free to drop your questions into the Q&A box at any point during the webinar, so you don't need to hold on to them. And we'll get to as many as we can at the end, so please ask as they come to you. Today, I'm really pleased to be joined by Aurélie Fougon from Mindpeak. Mindpeak is the leading company in AI-powered digital pathology, bridging the gap from biomarker development to clinical diagnostics. Aurélie is the Vice President of Predictive AI Solutions at Mindpeak, where she leads the strategy to shift pathology from descriptive to predictive approaches, supporting therapy response identification, treatment decision-making, and accelerated drug development. She brings over 15 years of experience across AI-driven precision medicine, biomarkers, and commercial strategy, with senior roles at Alkin, HalioDx, a Veracyte company, and QIAGEN. Her background combines deep expertise in biomarkers, pharma business development, and product strategy with a strong ability to translate complex scientific and AI concepts into clear business and clinical value propositions. So today, let me give you a quick overview of where we're headed. So Aurélie will cover what they're building today at Mindpeak and how they're making their algorithms accessible to you. Then we will finish with your questions. So again, put those in the Q&A box as we go. And before I hand it over, I'll spend around 30 seconds on who we are for anyone who is new to Techcyte today. So Techcyte is a digital pathology company. We're founded in 2013, and our digital pathology platform Fusion supports both anatomic and clinical pathology workflows. We're also in the veterinary and environmental space as well. There are more than 140 of us now, and we are a remote-first company with team members spreading across 27 states and 5 countries, with offices in Orem, Utah, and Luxembourg. We're in more than 40 countries, and we've passed over 10,000 customers, and we've now delivered over 10 million AI-assisted reads through the platform as well. One honest note on regulatory status, because it really matters in this field. Fusion is CE marked in the EU and UKCA certified in the UK, with a range of our suites covered under those frameworks. In the US, the platform is currently for research use only, and I can take any of the specifics in the Q&A. With that, we are ready to turn this over to Aurélie. So please go ahead, Aurélie. Thank you so much.
Thank you very much, Tiffany, for the nice introduction. It's really a pleasure to be there today to try to talk about Beyond the Viewer and how Mindpeak is here today to help and support pathologists in their daily routine. So for that, I think as a pathologist, you all know that you need to zoom into hundreds of images per day. You need to go in very details, look about slide, about tissue, also about cells, even inside the cells. And this, of course, takes time, and it's very important because you need to interpret this complex signal to be able at the end to give the right diagnostic to the right patient. But this detection is only the first step, and you need to understand and to count the number of positive tumor cells, the number of negative tumor cells. You need to know if you have an immune infiltration, if T cells or B cells are there, and this is very important for you. And small difference can change outcome. It's a high resolution signal that drive at the end your high-stake decision and give the diagnostic that is so important for our patient. And I think we all know today that this system is under pressure. The traditional pathology need interpretation by human, and it takes time. And we have more and more shortage of pathologists in many regions. And to have this traditional pathology, you need to have a lot of interpretation. You need to take a lot of time because tissue complexity is even more and more exceed, and it's really combine that with an accurately quantifying approach that is more and more complicated. And at the end, precision medicine that is so important for our patient demand a higher dimensional and multimodal biomarker. And today, because of all of that, we believe that the human eyes can't do it alone. it's not about replacing human, it's to help them to be augmented, and at the end, to understand the complex biology that is behind and really answer the complex question. Because it's not a clean lab problem. It's noisy data, high variability, and real-world constraints where mistakes have consequence. And this chart shows an example of a Ki-67 biomarker in 3 different pathology labs in Sweden. Same disease area, same biomarker, large sample size, and what happened at the end, the way the median result is materially different. And in some labs, you can be eligible to a treatment, and in another, you can't be. And it's not about the biology that change, and it's not about a problem of pathologists. It's just because complex visual pattern, it's difficult to assess when you are under pressure and under time pressure. And we truly believe at Mindpeak, this is what exactly the kind of problem where AI added value, and to not against replacing experts, but to help them to standardize the measurement at scale. So for us, we envision a world where tissue AI biomarker enables to new standard of personalized patient care. We truly believe that in the future, AI will unlock biomarker and spatial dimension and really reveal the tissue signal and give you and helps you to improve patient outcome. And to do that, you need to have the lab of the future. So you need to have tool that is today integrated into your routine daily practice. You need to have a way to interpret slide in a precise and reproducible manner. You need to have it in your classical workflow and not to a fancy place where it's difficult to get access to. And it's only because you will have the good biomarker and the good lab that we will be able to enter to the new era in precision medicine. So to talk a little about Mindpeak, we are a German company founded in 1988, 18. We have above 20 tools today on the market, CE marked and also used in the US thanks to integration with key partners. We have tested more than 80,000 patients, and we are able to integrate our system in a lot of different IMS like Techcyte. And we are very proud to be integrated with Techcyte and to be able to, at the end, give to all pathologists the capabilities to have access to AI. Today, we also work with pharma, CRO, and diagnostic lab on different projects, and we really try to push as much as possible AI tissue biomarker to the next steps. But before going even to an AI tool, what is important to understand first is to go back to what is the core capabilities behind an AI tool. And for that, we need to talk about foundation or frontier model. That is the beginning. It's very important to take time to understand that, because to have a good AI model that support routine test diagnostic, you need to have a stronger foundation because this foundation will be the one that will extract tissue, segment it, extract cells, classify them, and then be able to understand even inside the cells what is happening. And with a foundation models, you enables you to achieve superior performance. You accelerate all other AI development timelines, and you enables model training with limited data. So it's very important, and it's what Mindpeak has developed since 7 years. We are continuing and continuously upgrade our models with always modalities that is image, MIF, IHC, H&E. We have above 5 million expert annotation that help us to, at the end, build a frontier model that is pan-tumor coverage with 12 different tissue region type and 13 different type of cell. And it's because you have that as unique breadth of insight that you can then go inside the tissue and understand what is behind and go to the next step. Our peak frontier is really the basic of our unique technology. Then we combine unsupervised and supervised learning methodology to be able to go at a cellular and even subcellular precision. it's important to combine the different methodology, to combine the different kind of image and different modality to be able then to really match robustness and understand how you can at scale analyze pan-tumor tissue and find downstream task that is biomarker. If you have a strong foundation model, then you can ask him to look at specific biomarker to help you to count cells, to help you to subdivide the cell into cytoplasm versus membrane. And it's because of that that we are able to develop the next generation of AI tools that support pathologists. Because at the end, the objective is really to standardize measurement with AI. We want to increase accuracy and find previously hidden information to support pathologists on their daily routine. And so our solution, it's called PeakDX. It's really a way to turnkey analysis of IHC because we capture, thanks for example to Techcyte Fusion, the IHC slide, and then we decode it with our AI solution. The AI solution is running directly with zero-click as a backend and directly give you the information. And this information then it's analyzed, and you will have the chance to see a real demo at the end of this presentation to show you how we analyze the data. And it's really something simple to use it, and you see the TPS score, a CPS score, or any score that you think it's important, you can have it on this solution. Our group of clinical grade diagnostic AI tool cover breast suite, pan-tumor HER2 suite, pan-tumor PD-L1 suite, and other additional solution. On the breast suite is really important for us to go to all the classical biomarker. So HER2, PD-L1, K67, ER, PR, so you can directly, from your IHC image, calculate the biomarker and understand precisely the analysis around the entire whole slide images. We have the same with a focus on HER2. We all know that HER2 is very important biomarker, not only in breast, it's also important in gastric, lung, and so we have pushed the models to be able to analyze in a pan-tumor settings. Same for PD-L1. Today, PD-L1 is a very important biomarker in daily routine and makes the TPS and CPS score, the H score, depending on the cancer type, it's always complicated. The AI solution is here to analyze at the single cell level and really give you a standardized quantification across every tumor type. And at the end, we also provide prostate analysis directly from H&E with the fact that we have the capabilities to measure between MIF, IHC, and H&E. We are really able to propose other solutions like also quantifying the TILs, the tumor infiltrating lymphocyte, and develop other biomarker that can be used during, for example, also clinical trial, when you also need to, at the end, a pathologist to analyze it. So really, our objective is to streamline your routine works with Tissue AI Suite and at the end, augment your way to do. We try to reduce anti-reader variability. We also reduce your workload. We strengthen the endpoint reliability. And we, of course, standardize the analysis across site because as you use the same tool at the end, you harmonize the site performance globally. It's very important for us to automatize and automate all this pathology workflow. That's why you integrate solution, because having a good AI tool, it's a good first step, but being able to integrate it in your daily routine, it's where it makes even more sense and give you faster readout and turnaround time reduce, which is so important in routine. And of course, we scales because expand the reach with it's really important by pathologist headcount, and using AI tool can really support this part. And real examples before going to the demo, the live demo, is just to show you, to conclude, 2 projects we did. This first one we did with the Mayo Clinic, the MD Anderson Cancer Center, John Hopkins, Memorial Sloan Kettering Cancer Center, and BMS and Emory University. This was really for objective to transform the manual CPS scoring on a whole slide imaging with something automatized. We all know that is really time consuming, and so we have developed the AI PDL1 tool to really measure in gastric cancer this test. What was really important, and we present that recent years in different conferences, is that at the end, we really increase inter-reader concordance from 43% to 69%. And at the end, we were even able to identify 50% more patients that are at the end really eligible for IO therapy. And this is where really AI can help and can be put in routine today thanks to Techcyte integration, because you can use this test in your daily routine. And same for HER2 scoring. We present that at ASCO in partnership with AstraZeneca. Again, analyze HER2 scoring, different trial sites, and it's really long, and the inter-reader concordance was difficult. So at the end, we deploy our solution in 10 different countries. More than 100 pathologists was using our tool and tested in more than 2,000 patients. And what was very important is that with our solution, we were able significantly boost inter-reader concordance from 1 in 3 to 1 in 5, and also to improve the accuracy in ultra-low and low HER2 expression range. And at the end, we reduce the number of patients missed for 30.5% to 4.5%. So definitely using an AI component in HER2 scoring helps really inter-reader concordance and scoring accuracy for your interpretation, especially on the ultra-low sample. So I hope that we all here to know that AI is important in pathologist workflow, and tool is working now. Tool can be displayed and deployed in and integrate in classical IMS. And thanks to Techcyte, we can really be able to use every day in your routine work this kind of tool. So I would like to, of course, thank you for this time, and I hope you will have plenty of questions. And I will have Tiffany back now to show you in real how it works inside Techcyte. Thank you.
Well, thank you so much, Aurélie, for the very comprehensive overview of Peak Frontier and Peak TX. That was just very cool to learn about your diagnostic AI portfolio and how your solutions can really help improve concordance and accuracy in IHC interpretation. So next, we're really excited to show you exactly how this IHC AI can be deeply integrated in the Techcyte Fusion platform to help support your day-to-day workflows. So in this demo today, we'll be showing two cases launched from the Techcyte worklist and in our viewer with the Mindpeak algorithms applied. The first one is a breast core biopsy. So as we look at this core, we see that there's invasive ductal carcinoma present. Typical workflow is you go on to order ER, PR, HER2, Ki67, and then reviewing those slides can be a bit tedious. So this is really where Mindpeak's algorithms come to play. So the first stain I'll look at is Ki67. This is a 4-region algorithm, which means, like its name implies, that there are 4 distinct regions where the algorithm is applied on. And you can jump to these regions using the lower right-hand side widget built in. So coming to one of these regions, you can see which cells are marked positive and they're dotted in red, and which cells are marked negative, which means they're dotted in yellow. You can also adjust the positivity threshold. You can lower it, raise the threshold, and you'll see the results change accordingly. And then from here, you can also minimize this widget. So when you minimize it, you basically remove the annotations from the screen so you can view the slide without the AI applied as well. For PR, this is applied on the entire core biopsy. I see a mix of red and yellow, which means a mix of positive and negative. So upon higher magnification, you can also see here, some of these cells might fall on the border of positive and negative, and so we allow you to also use the positivity threshold to adjust where you believe your cutoff is. And like KS67, you can also minimize the widget so you can view the entire slide without the algorithm applied as well. ER is going to be very similar in this case. ER is more strongly positive. You see a lot of red. So then you'll see here, maybe there's not as much need to adjust your positivity threshold. You can look at it at a higher magnification and be like, yes, these all look strongly positive to me. I do agree with this call. No need to adjust the threshold here. For HER2, it's a little bit more nuanced. HER2 scoring is more complicated in the sense that you're no longer reporting on just positive and negative. You have tumor negative, tumor partial, tumor complete strong, and tumor complete weak. And you might want to view this first without the algorithm applied. You can see here there's strong membrane staining, and then you can toggle on the algorithm to see if you agree. And the algorithm has called it 3+, so predominantly strong complete membrane staining. So it's just really helpful to have these algorithms present for these cases, especially as I review, many different IHCs at once. And it's really nice to have the built-in AI results widget that we have purpose built for these algorithms. So the next case I wanted to go over is a lung PDL1 case. As we know, Mindpeak has many different algorithms, and this is just these are just a couple of them that we're showing today. For this lung PDL1, it's very similar to the breast workflow where different cells are marked with different colors, and you can always toggle on and off the AI results. So in this case, maybe I want to review this first without any AI results to get a general feel of what this PDL1 stain looks like. And as I toggle it on, I can see, do I agree if these tumor cells are positive, if it's tumor negative, or if these mononuclear inflammatory cells are positive here. So hopefully this gives you a good overview of what Mindpeak algorithms look like inside the Techcyte viewer. Again, Mindpeak has many different algorithms, and if you're interested in learning more, please reach out. Hopefully you get a sense of what deeply integrated AI looks like in our Techcyte Fusion platform. And with that, we'll turn it over for any questions that you may have. And again, Mindpeak has many different algorithms, and Techcyte as well. We're able to support those. So please reach out if there's a specific algorithm that interests you today. All right, so it looks like we have a couple questions. So the first question we have is from Peter. He asks, is it one single algorithm that covers every organ type, or do you need a separate model trained for each tissue type? I can turn that over to Aurelie to respond.
Yes, of course. Thank you. Thank you for this question. So yes, as I was presenting, we use Peak Frontier as the same AI backbone for all indication. And this is very important for us because then we can push our models to then be pan-tumor HER2 or PDL1, but at the end, it's always only one model that is running across tissue type. And it's really valid. Then it's validated separately per indication, but rather to build a separated algorithm. So it's very important for us to use this strong Peak Frontier model as backbone to be able then to move to the next step. And so, yeah, it's only using this one, and then we fine tune it with our PeakDX solution.
Great, thank you so much. And then Ryan asked, does implementing AI require us to standardize all of our staining protocols first?
No, again, it's really important. Mindpeak has worked a lot on also integrating different antibodies, different methodology, different workflow. Of course, when we implement a tool, we need to do calibration with 5 to 10 slide maximum, where we can, and as you also shown, Tiffany, we have the abilities to, make some your own modification. Again, the pathologist is still at the center of this analysis. And so the more you know on the workflow, then the better. When we implement it, we can check. But really, Mindpeak was also developing, again, thanks to this Peak Frontier model, we are really able to integrate a lot of different antibodies, IHC testing, and methodology. And this is what makes stronger the model also at the end, because then you use in the same way for the same in your routine workflow. You don't need to change anything. The AI come on top.
Great. And then maybe to segue, Lisa asked, how do pathologists maintain control over the final diagnosis while benefiting from AI assistance? They already just mentioned, we did see the positivity threshold. So you as the pathologist, you still get to determine where your cutoff is. And then from the Fusion perspective, the results are shown in that widget or in the AI results widget that was at the corner of the screen. You as a pathologist can still go in and decide whether or not you want to add that to your report. So you're still in full control there.
Yes, fully agree with you.
And then we have another question from Maria. She asked, for validation on our end, roughly how many slides would we need to run through the system?
Yeah, thank you, Maria, for this question. I briefly touch on it, but yeah, we roughly ask for 5 to 10 slides per biomarker for calibration, because it's always very important to still have a look with our partners and with lab, but we don't need more. And then we, of course, check with the pathologist and with the lab together to see if it's good. Of course, if it's more for, I would say, not a PICDX tools, and it's more a biomarker that is under development for clinical validation, we must turn around 50 to 60 cases to really be able to validate the tool to use it for, specific clinical trial or things like that. But really, it's a low amount of slide to be used just to be able to, at the end, integrate the tool in classical routine.
Thank you. And then Katya asked, does the analysis kick off automatically before the pathologist even opens the case?
So yes, it's true that the integration into the Techcyte Fusion system did the way that the AI model fuel inside the IMS when the case is uploaded and directly, analyze it, what we call the zero click by default. But at the end, the IMS and the pathologist remain entirely under control. So when, and you show it also, Tiffany, we are able to just have a look on the IHC image without any AI on it, and then you just click and the AI is coming. He has done the calculation before, but then the calculation, you can also choose your region of interest, you can make your own modification, you also touch about it, and then you validate. And when it's validated, it's done. I think it's very important to also really, when we talk about having to reduce turnaround time, is that why it's important that the model is behind? Because if you click and then you need to wait that the model is coming, that's not helpful. But then you still stay under fully control of what is the result that you want to provide at the end. So it's a way Mindpeak works, yes.
Great, thank you. And then with Michelle, she asked, how do the results and reports actually flow back into our LIS? And what does that mapping and integration look like in practice?
Yeah, so we have automated report, PDF report that is generated when the pathologist agreed on the methodology. We can also have surface chip and endpoint in the LIS for triage, and so it's possible to make some customization depending on the need. You can have individual slide analysis or case analysis. You can do other report on that. This is also, again, we have automatize as much as possible, especially the report analysis, but we know that workflow can be different from lab to lab. And then it's also a question that we have together with Techcyte, Mindpeak, and the labs to discuss and to see how is the best option to be integrated, and then we can do and modify it. So that is roughly our methods to do the reporting and then to integrate with the system that is already in place.
Great, thank you. And then we have another question from Simon on the topic of reporting. Can the AI be customized to match our lab's reporting preferences?
But yes, so I briefly touched on it also just before, but yes, it's of course it's took a little more time on the integration process, but it's something that we truly believe it's important, and so we can discuss with the lab and with you Techcyte to be able to then make some modification and to integrate that. It's really because our AI is done as backup that it's possible to still make some connection, and yes, it's what we provide based on question and needs that the labs can have. Yes.
Wonderful. So maybe just one more question then for today. So this is from Ruth. For HER2 IHC AI analysis, what magnification works best? Also, is it necessary to use layer by layer scanning like Z stacking?
No, I don't feel. Of course, with the 40x magnification and 20x magnification, it's the 20x is the classical way to do, and then you can do more. But also, same. We have work when we have developed the test in different magnification layer, and also we have used different image analysis to be able to perform. So we have some recommendation to follow the CE-IVD version of our tool. But then it's also a discussion that we can have based on the workflow of the lab, and we can work on it. I think it's also more the pathologies that has their own use to, and you don't need to go to 40x magnifications to be able to use our AI. This is definitely not needed.
Okay, great. Thank you so much. And thank you everyone for all these questions. With that, we'll end this today. Thank you, Aurélie, for coming and presenting on what you're developing at Mindpeak. We're super excited to be partnered together and to bring this IHC AI to all the pathologists here today. So thank you so much.
Thank you so much also for being present today and for this time. It was really great. Thank you again. Have a lovely day, everybody.
Thank you. And just one more thing, this webinar is recorded, so we will be posting the recorded version of the webinar. So please follow us along. You'll be able to see when this gets posted on LinkedIn. And thank you so much again for all of your time today.
Thank you. Bye.